Hello everyone, so my name is Nurhafiza and this is the second lecture for MIC3011 of the Advances in Medical Virology. I'm going to talk about pathogenesis of viral infections. So as everyone knows, UM is asking us to do e-learning instead of the face-to-face lectures. So this is how I'm going to do my lecture. So if you have any questions or anything that's not clear, you can always email me. Yeah? So this is the outline for this lecture. I'm going to start with introductions, and then the portal entry of the virus, mechanism the virus spread in the host, the factors involved in viral pathogenesis, and then the effect of the virus infection on host. So what is viral pathogenesis? Yeah? It's actually a process by which a viral infection leads to disease. Not all virus infection can cause a disease. Yeah? Some of the virus just get into your body and then your body responds to it and then produce immune response and then kill the virus. Yeah? But some of the virus can cause the disease by certain mechanism that we're going to talk later. So there are basic requirements for the virus to cause infections. First is it needs a sufficient virus to initiate infections. It's more on the amount of the virus, okay, that enter the host. And then the second one is the cells at the site of infection are susceptible and permissive. Yeah, so we're going to talk more on the meaning of the susceptible and the permissive later. Yeah, and the last one is the local host antiviral defense system is ineffective or absent. So these are the mechanism of the virus pathogenesis. Yeah, they usually start with the portal entry of the virus. Yeah, the virus enter the host, and then it causes a local infection of the virus. Yeah, some of it will spread to the target organ, and then it will go. It will be shed. Yeah, the virus shedding, and that means that it will goes out from the host and then transmitted to the other host through transmissions. Yeah, we call it transmissions of the virus into the other susceptible host. Yeah. So actually virus, it is not in the interest of the virus to severely harm or kill the host. So because you imagine virus, being in the virus, logically if they kill the host, and if it needs a new host, which is very troublesome, right? So actually the effect of the virus pathogenesis or the virus disease, it depends on the interplay between the host and also the viral factor. So it's both viral factor and host factor that causing the effect of the infections, virus infections. So to study the viral pathogenesis, yeah, especially in the human virus, we cannot use human. Yeah. Definitely no. But then what we can rely on is the animal models. Animal model is used, but it was actually not absolute predictor. It's not really a mimic of what happened in the human. It can be used as a good reference, yeah, as a start, a good start, but not exactly what's going on in the human. Yeah. So the animal models that we usually use is mouse, rat, marmoset, macaque, fruit fly, and also zebrafish. Yeah. So these are the example of the small rodents and the large mammals. So the small rodents usually mice, rat, guinea pig. Yeah, and the large mammal is the non-human primate, dog, and also pig. Yeah, so there's are advantages and limitations of both, of either small rodents and large mammals. So for small rodents, there are a large number of transgenic models that is available that you can always buy online. Yeah, and then you can find online, yeah, and you can buy it, and then you can have a high number of animal per experimental group, yeah, because it's probably cheaper, and then easier to manage. Yeah. And then, but the limitation is that the anatomy and the physiology is quite far, I think, yeah, than the human. And then for the large mammals, the advantages is because the anatomy and the physiological is much closer, yeah, to the human features. And then the limitation is the ethical considerations, yeah, and then financial, it's going to be very expensive when you're using large mammals, and then also zootechnical issues, yeah, how to manage these animals. It will be much harder than the small rodents. Okay, And then another problem with the animal model is that they are not all permissive and susceptible to the human virus. Yeah? So what they did is they developed animal models adapted virus. So what they did is they infect the host, yeah, or the animal models for certain periods until it become permissive and susceptible. Yeah. Alright, and then the second one is transgenic animal models. As you can see here, transgenic animal models is meant when they add the foreign genetic informations into the animal models. So, for example, they develop a mice synthesizing human receptors. So they add the foreign genetics of the human receptors into the mice, and then the mice now can express the human receptor. Yeah? And then some of it causing the mice to carry the complete viral genomes, and some of it cause the mice to express an individual viral genes. For example, if in dengue you want the mouse to produce NS1 protein of the dengue virus, then they will induce them. They can actually do that. Yeah. So now the virus can express NS1 continuously in the body. Transgenic animal models, from not able to do that to able to do that. Yeah, so we can do that in the mouse. And then some of the animal models, what they did is they do the human organ allograft animal models. So they just they put the human organ in the animals, so that now the animal can actually produce, can actually react like humans when they were infected by the virus. Yeah. And then some of it virus expresses transgene in live animals. So this is the one that they mentioned, mice expressing individual viral genes. Yeah, so they can express that continuously. Yeah. So this is how they actually to produce a human animal models. Yeah, humanized animal models. So this is what they did. Yeah. Make it more human. Yeah? Make it not entirely as a human, but a bit like a human, so that you can actually have an idea when the virus infects the mice, probably it's the same thing that happened in the human. Right? Okay, so other methods for the analysis or the study of the virus pathogenesis is cell culture also we can do. Yeah, and we can look at the epidemiology, yeah, and also the viral genetics. Right? So viral genetics also play a role in the development of the disease. Okay. Alright, so we start with the port of entry of the virus. So there's actually three main sites for the virus entry. So the first one is inoculations. We call it inoculations, are more like introductions. Yeah? Inoculation of the virus. It has under inoculation there's two, yeah, via the skin and also mucosa. So via the skin is an effective barrier against the viral infections because it's a dead outer layer, cannot support the viral growth. So if it's there, then it will die because it cannot support. Yeah? But then if there's a breach of skin integrity, such as accidental abrasions, a needle stick injury, or by the bites of the arthropod vectors such as mosquitoes or ticks, yeah, then it can actually cause infections, yeah, when the virus can enter through the breach of the skin of these two methods. So once the virus reaches the dermis and the hypodermis, yeah, it has access to the blood and the lymphatic vessels, so it can cause the spread of the infections. So you know that your dermis, this is epidermis, keratinocytes. So when it was on here, it was nothing happened. Yeah, they cannot cause an infection because they cannot get in. Yeah, and then the cells there is dead, so cannot support the viral growth. But then when it's get in because of the injury, yeah, or when there's a virus, or when there are mosquitoes that trying to try to feed on you, yeah, then it can cause a breach of the skin. and can cause the transfer of the virus. And then when it gets into the blood vessels or lymphatic vessels, it can be spread. Yeah, of course, there's a natural defense mechanism, innate immune response. For example, macrophages or even lymphocytes. But then some of the virus can evade this kind of mechanism, the host defense, and then cause a further infection and spread. So this is by the skin. The second one is via the mucosa. So virus transmitted directly, remember, directly when mucous membrane, which is the thin moist lining of many parts of the body such as nose, mouth, throat and genitals, comes into contact with the skin or mucous membrane of another person. So when the mucus meet with the mucus of other person, it causes direct transmissions of the virus. But then it will be caused indirectly when the mucous membrane comes in contact with the contaminated objects or surfaces. Okay, so not mucus surface, but the contaminated objects or surfaces. For example of the mucosa, side of the mucosa is the eyes, oropharynx, tract, basically our pharynx, and then respiratory tract, intestinal tract, and also genitourinary tract, which is the urogenital tract. So these are where the mucosa is. And then of course there's a natural defense mechanism. We have a mucosa secretion. So the mucosa itself has the antibody, which is the IgA, that can actually neutralize the virus. And then they also have a mechanical and chemical defense in the mucosa. So mechanical is usually the cilia. They have the cilia there, mucosa itself, right? And the cilia, the movement of cilia usually prevent any attachment of the virus or even other pathogens. The chemical defense, which is the mucosa itself, have the antibody, and then immune mechanisms, other immune mechanism around the lymphocytes or even phagocytes. And again, I just want to emphasize that even though with all of the defense, some of the virus can still evade the host defense and then cause infection and spread. So this is inoculation, it's the first one. So the second one is the inhalations, which is via the respiratory tract. To obtain access to respiratory tract, the virus need to be primarily in the form of aerosol, particle or droplets. So usually when you cough or when you sneeze, then you produce an aerosols or droplets containing the virus if you are infected. So the factors that affect viral respiratory infection is humidity, air temperature, the physical, chemical properties and structure of the virus. Okay, so there are also defense mechanisms for the viral entry through the respiratory tract, which is the same, so you have the mucus, and then action of the cilia epithelial cells. So this is the mechanical defense. The IgA present in the respiratory secretion, which is the mucus, antibody, and then alveolar phagocytic cells. So this is the phagocytes, innate immune response around the alveolar, and then of course the other cellular and humoral immune responses. Okay, so same with just now, some of the virus can evade all of this and then cause an infection and disease. Okay, so this is the close up from here. So this is the larger image for this part. So as you can see here, there's a mucus layer, first a physical barrier for the virus to get in, but then they can still get in. And then ciliated cells, you got cilia there to prevent any bindings. But then some still can cause infections. So this virus above showing the virus that can cause infection on the upper respiratory tract, near to the nose too. And then these are those viruses that can cause the infections on the bottom and near to the alveolar, the respiratory tract and the alveolar. As you can see, this is a macrophage, the defense mechanism in the respiratory tract. And then the third pathway is the ingestions via the gastrointestinal tract. So the ingestions of water or a wide variety of foods contaminated with disease-causing germs or their toxins. If you ingest anything that contaminated, it can cause infections. We call it fecal-oral routes. Fecal-oral routes means the fecal particles pass from one person to the mouth of another person. So it's quite it seems like impossible, right? It's like, how can something like feces get into your mouth? But it actually can, if you have a poor hygiene or poor hand washing hygiene methods. If you will not practice a good hygiene, it's possible to get feces, particle of it. It's like you will not eat the feces, right? But the particle is eaten on your hands, and then you don't wash your hands and you eat. That's when it can get into your mouth and then cause infections. So it's very important to have a good hygiene, especially the hand washing, hand washing hygiene. Okay, and then of course the same like the others pathway, they have the defense mechanisms. Continuous desquamation of the epithelium. Desquamation means like shedding off of the cells. So if the virus is at the epithelium, and then because of it continuously being shedding off, and then it will die, right? So it will die together. The virus will die together with the dead cells. And then this is more mechanical. And then this is the chemical: saliva. We have mucus layer of intestine, gastric acid, bile, and proteolytic enzymes. So these are the defense mechanisms of the host against not only virus, other pathogen as well. And then mechanical movement of the tongue, the cheek, the peristalsis, it does play a role. And then of course the host immune mechanisms. So virus that infect by this pathway or route must at least resistance to extreme pH, because we have an extreme pH in our stomach. We have enzymes, proteases, and also bile. So those that can resist all of this usually able to cause infections through this pathway, ingestions pathway. So this is just want to show you where is it, the microvirus. So this is the cilia, and then we have macrophage, lymphocytes, which is there to actually kill the virus, but then some of it can evade, cause an infection and be spread through this entry. Okay, so these are the examples of the viruses with their specific routes. But some of the virus can cause more, can get into the host through more than one sites. For example, HSV, it can go to the urogenital tract and then also to the eyes, the mucosa of the eyes. So it will be good if you can remember some of it, and its specific route. Okay. So now we will go to the mechanism of the virus spread in host. So these are the mechanism, like I mentioned earlier. It starts with the direct locals, lymphatic, viremic, and then the CNS. Some of it only local. Some of it go to lymphatic and stop there. Some of it go to viremic, stop there, or some of it spread to the CNS. So some of the virus can cause local only, and then probably go to lymphatic, but then stop there, and then some can evade the thing in lymphatic and then go to viremic, and then some can spread to until the CNS. That's it. So the local spread is when the new release virus infects the new ring cells, adjacent cells, and then the infection contained by the physical constraint and controlled by the immune response. Physical constraint means possibly because of the, if there's a mucosa there, it stop. And then if there's a cilia, cannot bind to it. and if it's in fact in the keratinocytes, whereas usually the dead cells, it cannot support the viral replication, so it's a physical constraint there. And then some of it, not only physical constraint, but it also controlled by the immune response. Yeah. For example, like once overcoming the epithelial barrier, the second line of the body defense kills the virus. So it's like there only, it's local. Yeah? Infection without the systemic spread. And then usually the virus that spread beyond the primary sites, we call the process the disseminations. Disseminations. And then if they can evade the immune response and the physical constraint, it can be spread to the lymphatic vessels, lymphatic capillaries, as you can see here. You can go to the lymph node which is nearing to the infection sites. Yeah, for example, I mentioned in the skin, right? If it goes to the dermis or epidermis of the hypodermis, it has the lymph capillary and also the blood vessels there. So when it goes to the lymphatic capillary, it will carry it to the regional lymph nodes. Yeah. So function of the lymph nodes usually filter the microbes. Why they say filter the microbe? Because it is a site where immune response generated. So there's a lot of phagocytes or lymphocytes there. So usually if the virus is exposed to these kind of immune cells, phagocytes and lymphocytes, the infection will resolve. But some of the virus that can evade the immune response, what they did is they managed to hijack the cells, and causing the cells to not killing them. But not only not killing them, but they will enable the virus to replicate. They will allow the virus to replicate in them, in these macrophages or lymphocytes. So this virus and measles usually do this. And then this is not only increase the virus load and the inflammatory response, but at the same time they use this macrophage and lymphocyte as vehicles, because we know macrophage and lymphocyte usually circulate the body through the lymphatic and the blood vessels. So if they manage to causing these immune cells to not killing them and then allow them to be replicating, then they will act as a vehicle. So when they replicated, they will be at the same time will be transported throughout the body, and then they can infect the other cell as well. They can spread. They use the immune cells as vehicles, and possibly one of the reasons that the virus can enter the bloodstream. So for example here, the virus, some of it have a breach of the skin, breach of the mucosa or something, and then it can infect the cells, and then it goes to the lymphatic capillary nearing, and then from the lymphatic capillary it goes to the blood vessels. But some of the virus, if it's through arthropod or iatrogic, usually what happened when using the needles or something, right? So if it's contaminated, it can transfer straight to the vein, because usually if it's mosquitoes, if they want to feed on you, they will get into your blood vessels. If they can poke through and then transfer the virus there, they will go straight away to the blood. And the same with the needles. If they want to take your blood, and it was contaminated, and will transfer something else in your blood. So they have a few ways how to infect the blood vessels. So that's when they get into the blood vessels, and of course from the blood vessel it goes, all the way into your body. They call it viremia. So when it gets into your body, and subsequent spreading is called viremia. So we have two types of viremia. The primary one is the first one. It's the first episode of entry into the blood. Usually in the lower concentrations. And then it will go to second, viremia, which is second wave of viral entry into the bloodstream, infection of other organs. Usually it is high concentration of the virus. Yeah? If many organs are infected, then we call it systemic infections. Okay. Yeah, so this is the example. Passive viremia is actually the inoculation, the first one. It was not actively replicates, but when it started replicates, yeah, it will be like this. Primary viremia, but then on secondary viremia, it will be very high. Yeah. Okay, and then the last one, the mechanism, it can go to the, some of the virus can spread to the CNS or central nervous system, and peripheral nerve spread. So there are three ways how the virus can spread to CNS. The first one is neural. So we know that neurals is through the neurons, yeah? The neuron pathway. Usually it was used to send signals, yeah? So but then virus can actually go through this pathway to go to CNS. And then we have olfactory. So olfactory is near the nose, yeah? It's a nerve pathway for smelling, yeah? Near the nose. And then the last one is hematogenous, which is the blood vessel actually, yeah? Originating and carried by the blood. So it can go three ways to go to the CNS. So those who are from hematogenous, yeah, which is the blood vessels, during viremia, viruses invade the central nervous system by localizing in the blood vessels of the meninges and choroid plexus into the neural tissues. So these are the blood vessels nearing the spinal cord and the brain. Some of the virus, and then some of it just directly infect the brain through the spinal cord or brain. Yeah, so they just directly causing the infections in the brain. The second one is through the peripheral nerve or the neuron. Yeah, it is actually an effective path of transmission for some viruses. For example, herpes virus. Yeah. The mode of transport is a slow process compared to the viremic spread. So they have three just now, right? The hematogenous, the viremic, the blood vessels. So this transport method is much more slower. Yeah. So the routes of viral transmission is the axon. Yeah. Through the axon, there's a few routes they can go, and then endoneural space, the connective tissue space between the nerves. Yeah. And then the perineural lymphatic. Yeah, this side of the nerve. Yeah. Okay, and the last one is the olfactory routes. This is the one that I mentioned, the nerve to sense. Yeah, it's nearing. This is our nose. This is, where is it? This is arachnoid lah. It's the opening. This is where the virus can actually invade the brain through this opening called the arachnoid. Yeah. Okay. Alright, so virus shedding and transmission. Virus shedding is the release of infectious virus from the infected host. And there's a few sites of shedding, which is the respiratory secretions. So this is where the virus can goes out. Yeah, coughing, sneezing, and then saliva, feces, blood, urine, semen, and milk, and skin lesions. Yeah, and then it will be transmitted. Virus spread from one susceptible host to another. Okay, so it starts from the entry, and then spread, and then shedding, and also transmissions. So this is a full mechanism of the viral pathogenesis. Yeah? So factors in viral pathogenesis, there's a few. Yeah. Like I mentioned just now, it depends on the interplay of virus factor and the host factors, and that can cause the disease. The first one is virus virulence. Yeah, the ability of the virus to cause the disease. Yeah, different viruses have different virulence. Yeah. It depends on the dose, the virus strain, inoculation route, the host factor, and the toxic products. And the second one is the entry and dissemination to secondary sites. So where they get in and how they will spread is also play a role. Yeah? The outcome of the infection determined by the location of the infections. Yeah, it can play a role. right? This is same also, localization and the spread of the infection. So those who can actually cause infections in the local only, possibly not that bad, right? But then when it can spread, not only in the lymphatic, but also in the blood, then it's bad. Yeah, it can cause a systemic infections, and then if it cause an infection in CNS, then it will be worse, right? So it depends on the cause of infection and the entry and the disseminations. And then cell tissue tropism, the spectrum of the tissues infected by a virus. So I will talk more on tissue tropism after this. So it means that not all viruses can infect all tissues or cells. So this, the possibility to virus to cause infections, yeah? More tissue or cell tropism that can actually receive this virus will be more problematic, yeah, compared to those who are like specific only, yeah? And then immunopathology means that the host defense mechanism contribute to the disease development. So this is when I say the host plays a factor as well. Yeah, so when the virus get in, the body said, okay, we need immune response, but the immune response was too much, and then not only it killing the virus, but it also caused a problem to the host. Yeah, it probably caused damage to the endothelial cells, or maybe caused damage to the platelets, yeah. And then the next one is the immunosuppressions, yeah? The exploitation of the host immune system by the virus. So the virus actually hijack the cellular mechanism. Yeah, they make the body to produce less immune response. Yeah? So when it become less, it will be harder to kill the virus, one, and then not only that, it will make the host become susceptible to other infections, fungus infection or bacterial infections, that causing the disease to become much more worse. For example, in HIV, yeah, the well-known one is HIV. And then the virus clearance and persistence is also a factor. Majority virus infections are cleared, but some viruses may cause persistent infections. Yeah, they are true latency. They remain completely latent, such as herpes simplex virus, so HSV and VZV, varicella zoster virus. So when you answer, please put the full name. You do not put HSV or VZV. I don't have space here, but please remember, when you are answering the question, put the full name of the virus. Yeah? And then persistence that replicates continuously. When you say latent, when it is mean that it's not actively replicating, they're just there. Yeah, it was not detected. But then some of the virus can cause persistence, yeah, replicates continuously at very low levels, such as HIV. Yeah. Alright, okay, so we're going to talk more on tropisms. Yeah, so tropism is the spectrum of tissues infected by a virus. Yeah, so there are certain viruses can only infect certain cells or tissues. So for example, enterotropic virus replicates in the gut, so they can only infect the guts. Yeah, whereas neurotropic virus replicates in the cell of the nervous systems. But some of the viruses are pantropic. Yeah? Pantropic means it can infect and replicate in many cells, many cell types and tissues. So the viral affinity for specific cells is determined by the cell receptor for the virus. So it's important with the receptor that it can recognize the thing is the virus is the pathogen, so the foreign entity, and then react to it. Yeah. So if there's no receptor, they cannot recognize this as a virus. Yeah. So they will do nothing. Yeah. So it needs cell receptor. And then ability of the cell to support viral replications. Some can have the receptor but are not able to support the viral replications. Yeah. And then physical barrier also play a role in the viral affinity. If there's a physical barrier such as mucosa or cilia, yeah, then it will cause the virus to be much harder to bind to the cells, yeah, and cause an infections. And the local environment, pH, oxygen, tension, enzymes, and non-specific factors also play a role. And then in the gastrointestinal tract, there's a digestive enzyme and bile, which can also affect the viral affinity to the cells. Okay, So this diagram shows explain a bit regarding the susceptibility and permissive. So for the A, which is susceptibility, is the receptor. So important, susceptibility is when the cell have the receptor and able to receive the or recognize the virus. Yeah. And then permissive is when it can support the viral applications. Yeah, it can support the viral applications. The C is accessibility. Yeah, when there's a barriers, it cannot access. So when there's no barriers, or when the barriers is breached, yeah, like the skin, yeah, so it can access the cells and then infect the cells. Yeah. And then G is the host intrinsic and innate defense. Yeah? So when there's an innate immune response, yeah, from the host, it's either like if it's antibody, it will neutralize the virus, and then if it's like T cells, it can kill the virus or kill the virus infected cells. Yeah? Okay, so the viral virulence, a little bit more information on that. It actually depends on the viral gene products. Yeah. Those who are, some of it can alter the ability of the virus to replicate. Yeah, and then some of the gene product can modify the host defense mechanism. It can hijack the cellular mechanism of the host, yeah, and then modify it. Yeah? Either make it less immune response or kill the immune cells, yeah, or maybe just induce more immune response but not working against the virus. Yeah, so there are ways that how they modify the host defense mechanisms to support their replications. Yeah, and then some of the gene products enable the virus to spread in the host. Yeah, probably it's hijack the cellular response of the immune cells. Yeah, not killing them, not eating them, but then allowing them to replicate. Yeah, something like that. Yeah, and then some of the virus gene product is toxic. Can cause toxic protein and then cause damage to the cells, to our cells, to the host or tissues. And then in viral virulence, there are also host factors. In the host factors, for example, age. Yeah, some age have a tendency to be infected. Yeah, some infections can cause a bad infections or disease in children compared to the adults. Yeah, some in the old people compared to the adult, yeah, some in adult compared to the children or even the old people. So it depends on the virus. Yeah, sometimes the age factor also play a role in the viral virulence. Yeah, and then another factor is like the host. Yeah, we have a lot of factors from the host actually, but I don't want to talk too much on it. Yeah, just want to let you know that it is not only the virus itself that causing the problem, but also the host. Yeah. For example, the host factor number 2 is host gene. Yeah. Some of the human individual have these CCR5 delta 32 mutations. So as we know that CCR5 is actually the receptor. HIV bind to the CCR5 too, and then it will be assessed. Yeah, it will get into the cells and then hijack the cells to produce. To cause and then modulate means like it regulates the immune response. It change the immune response to be favored to the HIV applications because of the CCR5 receptor. So they found that those who do not have the CCR5, when they say mutation, means that it was knockout. There was no CCR5 mutations. Yeah, so this human, those who do not produce CCR5 are resistant for HIV infections. So they said those people with this mutation are less likely to get HIV infections. Yeah, but those who are not having this mutation are more susceptible to get the infections. So our host also, our gene itself do play a role in the effect of the virus infections. Yeah. Okay, so for the HIV pathogenesis, yeah, in the whole, yeah, just to give any examples. So there are a few factors that play roles in HIV. Yeah. For first one, they can manipulate the immune cells. Yeah, they can infect them, yeah, instead of getting eaten. Yeah, and then dissemination was also quite spread, yeah. The spread is wide as well, from the lymph nodes to the varicella, and then it can also go to the brain, yeah. And then not only that, they have latent infections. They have ways how to stay in the body, yeah? Either latent or low level infections, right? And then another one is what they did is they caused the viral they caused the CD4 cells to lyse, yeah? So because of the cell CD4 cells were lysed, yeah, it becomes a severe CD4 cell loss. So there's immunosuppressions going on, right? And with immunosuppressions, there will be opportunistic infections, yeah? When we have less immune response, then we are more susceptible to other infections, yeah? Other pathogens. So there's a lot of factors that play a role, that are actually going on in the HIV pathogenesis, as you can see, right? And then in dengue was basically an immunopathology, yeah, because when original antigenic stains, because I don't want you to get confused, you don't have to remember everything. The one that I want you to know is that in dengue, we know that there's 4 serotypes, right? So people said that if you get infected by 4 serotypes, and then you recover, and you have the immune of dengue serotype 1, but then in second you got infected by other serotype, which is 2, 3 or 4, and then your body thought it was 1, they will produce the same amount of the immune response, the one that they thought it was dengue 1, yeah. But then the antibody that was produced, or the immune response that was produced, was not really good, yeah. It was inadequate in terms of the amount, yeah, and then it was not really ability was lost, yeah, and then it was like the antibody or the immune response that was produced was not efficient, yeah. When it's not efficient, the virus get higher, and then the immune response keep on producing something which is not good, yeah. And when there's a lot of immune response, we know that it's going to cause cytokine storm, yeah. There's a lot of immune response going on which is not helping, and then at the same time cause a problem into our body, yeah. It can affect our endothelial cells, it can kill our platelets, yeah, and then increase the vascular leakage, right? So molecular mimicry as well, the antibody not only recognize the virus, but it also can react with our platelets and endothelial cells, and then cause the vascular leakage. And this is what I want you to remember from all of this, is that it is when your body yourself, yeah, your immune response itself, that causing the problem in the infections, yeah. It's the immunopathology, yeah, in terms of the factors of the viral pathogenesis of dengue. Okay, for our next part, or the last one, is the effect of virus infection on host. So virus infection can cause then acute infections, yeah? We have acute and also persistent, yeah? For the acute, it occurs when a virus first infects the susceptible host. It's usually recovery with no residue effects, and usually is the infection resolved, yeah? For the persistent, yeah, it is when the infection is not cleared efficiently by the adaptive immune response. The virus particle may be produced continuously or intermittently for long periods. Yeah? This one is continuously or intermittently. Yeah? Sometimes it's up, sometimes it's not. Yeah? So the categories of persistent infection, we have latent, usually by HSV, viral nucleic acid integrated into the host DNA, yeah, and then it becomes undetectable during latency, so it cannot be detected. But then the infection will be pre-emerged when the host cell immunity is compromised. So when you have less immune response, yeah, when it was compromised, you have a disease or something of immunosuppression disease, then it will re-emerge. Or maybe when you are old, when your immune response was not really efficient, then the infection can emerge back, yeah. The second one is the chronic infections. For example, Hep B, yeah, the virus persists in the prolonged period. Host usually asymptomatic, yeah, because usually if you have an infection and if you have a symptom, you will go to the hospital, right, and then you will find out how to stop the viral infections. But then if you are asymptomatic, you do not have any symptom, you're just fine, healthy, and then you will not do anything, and then the virus was happily, you know, replicated in your body. Yeah. And we call those people who are asymptomatic and with these infections carriers. It can be a potential source of infection for others, right? And then virus continuously detectable. Okay. So, and then there's oncogenic infections. For example, Epstein-Barr virus, EBV, remember full name, yeah? And then the virus hijack the cellular pathway and promote the survival and proliferation of infected cells. Yeah, so not only it hijack the cellular pathway, yeah, and then they will continuously replicating. At the same time, they will proliferate. Yeah, the infected cell will be more and more. Yeah, it become an oncogenic infections. However, these are not happen always. Yeah, genetic and development factors are important in determining whether virus is oncogenic in a given host. Yeah, some people got EBV but not got oncogenic, but some got. Yeah, so it depends on genetic and also development factors. And the third one is the slow virus infections. For example, measles and HIV, but not all. Yes, some it's very rare. The occurrence is very rare. And then the incubation lasting months or years. So it incubate for a very long time, and then followed by the progressive disease. Yeah. Usually by the immune suppressions when you have less immune response, and then that's time when it emerges and become progressive and lethal sometimes. Yeah. Okay. Alright, so we're basically done. So this is just important in terminology that maybe you can look at it, right? The meaning of it. Okay. So these are the book. You can find it in library, I think, and do some additional readings. So again, if you have any questions or you need any assistance to understand this lecture, just email me and I will help you. Okay, alright, thank you.